Okay, so they've learned that there are 3 steps in the transcription process: initiation, elongation, and termination, where RNA polymerase reads DNA and builds the first RNA transcript called mRNA, or messenger RNA. However, write in your notes that most eukaryotic mRNAs aren't ready to be translated into a protein directly after being transcribed from DNA, because mRNA requires processing. Make sure you get that in your notes. Now, remember that transcription of RNA processing occurs in the nucleus, and that after this, the messenger RNA moves to the cytoplasm for translation. So get in your notes that before translation begins, the cell adds a protective cap to one end and a tail of A's, now you know what A's are now, they're adenines, to the other end. That these both function to protect the RNA from enzymes that would degrade. So make sure you get that in your notes. Also write in your notes that most of the genome, anytime we refer to a genome, it's all the genes an organism possesses. So humans have a genome, elephants have their own genome. All right? So most consists of a non-coding regions called introns, I-N-T-R-O-N-S, non-coding regions called introns, and we'll look at introns here pretty soon, which are non-coding regions that may have specific chromosomal functions or have regulatory purposes. Also write that introns also allow for alternative RNA splicing. Now, because of this, an RNA copy of a gene is converted into messenger RNA by, again, doing 2 things we need to get in your notes. First is to add protective bases to the ends. So, again, you're looking at a strand of it right here in this diagram. So, one of the modifications is to add now a protective base to the end, and you see those right here. These are the protective bases to the ends. And the 5' end receives a modified nucleotide cap, sorry, we can't talk this morning. And the 3' end gets what's called a poly A tail. So the 2 modifications, now you got to know these specifically, they will definitely be at least a multiple choice question on the test. So to the 5' end, you see it here, modified guanine nucleotide added to the 5' end. This is a protective cap. And you see guanine now here with 3 phosphates. And then they add what's called a poly A tail. And the reason it's called a poly A tail is there's many A's. Okay? It's this poly A tail. So 5' end, a guanine nucleotide added, which is a protective cap. And then the 3' end has a poly A tail. So make sure you, in fact, let's have you get this segment in your notes correctly labeled, because we're going to talk about these things eventually too. The protein coding segment, start codon, stop codon, etc. We're going to talk in more detail about those in the coming slides. So let's get this in your notes. Okay, now let's talk about the second modification that's made to this newly formed transcript, which is still pre-RNA. Write in your notes that this pre-RNA still contains transcripts of both introns and exons, and you see those in here. Here's the intron, the blue segment, and here's the exon. Now, this is on a DNA strand, and remember, all this RNA is doing is copying this, so it's copying the exons and introns right into its code as well. Now, this still contains transcripts of both introns and exons. Remember, introns are empty code sequences, not necessary. Wow, sorry about that. Because they're not necessary, they are spliced out, where exons are segments of the transcript that contain correct RNA coding for amino acid sequencing, and they are spliced together. So make sure you get that in your notes. So eventually what's going to happen is these blue intron sections, which are no longer necessary, are going to get spliced out. And what's left now are these exons, which are correct amino acid segment information that will then be joined together. And that's what you see right here. Now you see this kind of peach colored stuff. Those exons now are hooked together down here in a mature messenger RNA molecule. So a question likely on the AP exam is for you to distinguish between a pre-RNA molecule and a mature RNA molecule. And your answer would be that pre-RNAs have introns in them that have not yet been spliced out. And that a mature messenger RNA molecule has these exons spliced together. So get that in your notes as well right now. So again, to reiterate, once this process is completed, pre-mRNA then becomes a working molecule of messenger RNA that's ready to be transported into the cytoplasm for the next phase of protein synthesis, which we're about to talk about again. It's important to remember that we're exploring the process of getting the instructions to make a protein from the nucleus to the ribosome here. As of right now, that process is only half complete. We have instructions copied and into the cytoplasm now. Now remember, we've got to finish that process, which means get those instructions to the ribosome, and then the next step would be to get the raw materials, the amino acids, from the cytoplasm to the ribosome, so now we can actually make the protein. Okay, now a bit more about this splicing process. And remember, we're splicing out introns and splicing the exons together. As you might expect, there's an enzyme involved. Enzymes are the most important proteins in the body because they drive all the processes in an organism. And we're going to do a special section of this process just on enzymes because they're so important. Write in your notes that ribozymes, R-I-B-O-Z-Y-M-E-S, ribozymes, are the catalytic RNA molecule that function as enzymes and can splice RNA by removing introns and joining exons. Ribozymes. So ribozymes are the enzymes that splice out introns and splice the exons together. So make sure you get that in your notes, ribozymes and their function. Now, ribozymes are not the only enzymes that function to splice out introns and splice together exons. RNA splicing can also be carried out by things called spliceosomes. And you see the word right here, spliceosomes. So get that in your notes. Spliceosomes. Question for you. How is it possible that there are millions of different kinds of human antibodies when there's only about 30,000 genes in the body? Now this was a short answer question on the 210 AP exam. So the question is, how is it possible that there's millions of different types of human antibodies when there's only about 30,000 genes? So in your head you got to be thinking, all right, millions of different types of antibodies. How is that possible with 30,000 genes? Okay, the answer is alternative splicing. Write that in your notes. Alternative splicing. Now, write down that alternative splicing refers to the different ways the exons of a gene can be combined. And by combining them, they produce different forms of proteins within the same gene coding region. All right, so the answer is alternative splicing, which means exons can be spliced in different orders, I guess is what it really means. And as you, again, structure, function. if we now, instead of having exon 1 first, we have exon 2 first, we've changed the structure. And because we've changed the structure, yes, you're right, we're going to change the function. Now, alternative pre-mRNA splicing is an important mechanism for regulating gene expression in higher eukaryotes. Write that down. One of the themes in this class is genetic variation. What we mean by genetic variation is that we're varying now the combination or sequencing of the genes. And this is one of the ways that genetic variation is accomplished. It's a process now called alternative splicing. So make sure you get that in your notes. Now, in one of our later experiments, we're going to use this little circular piece of DNA, all right, to splice in a gene to make a bacteria glow. So this is one of the ways now that enzymes work, is they splice in and splice out segments of DNA and RNA. So make sure you get that in your notes.